So the way that we get ATP from glucose is by breaking the glucose down a little bit at a time and harvesting electrons from the glucose molecule. So what happens is we have glucose getting broken down and we transfer electrons to a coenzyme called NAD+. And I always like to say NAD+ is the electron bus. So it transfers electrons from those organic compounds, glucose and what's left of glucose, after we start breaking it down, to electron camp. That's where the electrons are going. They're going to electron camp. So they get on the electron bus, they go to electron camp. So when NAD plus accepts electrons, it turns into NADH. And it picks up 2 electrons plus a hydrogen, a proton. So it picks up 1 electron plus a complete hydrogen atom. Proton plus electron. So even though it's picking up that positive proton, it's picking up 2 electrons. So the net change to the charge is -1. So because NAD plus accepts electrons, it is an oxidizing agent. It takes electrons away. And we use those electrons eventually to synthesize ATP. And we'll get to that towards the end of cellular respiration. So here we've got NADH, which stands for nicotinamide adenine dinucleotide. So here you can see here's adenine, and here's a connection through 2 phosphate functional groups. And then here's nicotinamide, which is a nitrogenous base, but it's not one that we use in any of our nucleic acids. But when it is in the oxidized form, before it has accepted any electrons, there's a positive charge on this nitrogen in this ring here. And then we take 2 hydrogen atoms, 2 complete hydrogen atoms, proton plus electron. 2 of those electrons are going to reduce that nitrogen. 1 of the protons is going to glom on to that nicotinamide portion, and then the other proton is just going to be free. Okay, so we've got nicotinamide in the reduced form, which is NADH, and in the oxidized form, which is NAD+. So the reason that we need these electron shuttles, the electron bus, to move these electrons along is because we're trying to slowly break down the sugar to totally maximize the potential energy stored in those bonds. In an uncontrolled reaction, we could burn sugar. I could bring sugar into the lab and just burn it. Straight up burn it with a Bunsen burner or a match or whatever. And it'll burn. And it's a fuel that will keep fire going. Similarly, you could put a match in the gas tank of your car and release all the energy in that tank of gas really quickly. This is not recommended. Very much so. It's an uncontained release of energy and heat and light. So you don't want to do that. What you want to do is step on the gas a little bit at a time to get you where you're going and put that energy to use for you. So same thing with glucose. We want to release the energy a little bit at a time in a controlled release to produce as much ATP as we can. So here are the 3 steps of cellular respiration. And you absolutely have to know these 3 steps. Really 3.5 steps. Glycolysis, which in the previous version of the book they say this is color-coded as teal. Pyruvate oxidation and citric acid cycle, also known as Krebs cycle. which are color-coded salmon in color. And then oxidative phosphorylation, which is the third part, which is electron transport and chemiosmosis, color-coded violet. And I can't help but wonder if it was a coincidence that the names of these colors are also names of organisms. Here's a teal, here's a salmon, and here's some violets. So, what is important? Everything on this slide is important to you. Meaning I expect you to know all of the stuff on this slide. At the very least, this is what you need to know about cellular respiration. So the first part is this universal process called glycolysis, or sugar breaking, where glucose gets converted into two molecules of a substance called pyruvate. Pyruvate then gets oxidized, meaning we pull electrons off of it to form acetyl-CoA. Coenzyme A is what feeds into the citric acid cycle, and the citric acid cycle yields electrons that go into oxidative phosphorylation. So each of these steps produces some ATP, and they do it in different ways. So we have substrate level phosphorylation, which I'll get into in a minute, and oxidative phosphorylation, which I'll get into in a couple of minutes. So glycolysis occurs out in the cytoplasm, in the cytosol, and the rest of it takes place in the mitochondrion. So you're going to see why we call the mitochondrion the powerhouse of the cell. And given what I've been telling you in chapters 6, 7, and 8, we're going to find out why it's so important that these membranes are all folded up like this, and why and how the role that enzymes play in making all of this happen. Okay, so the process that makes the most ATP is that last step, oxidative phosphorylation, which is powered by redox reactions, moving electrons. Electrons get transferred from one molecule to the next, and we use that energy through electron transfer to produce ATP. So about 90% of the ATP produced by cellular respiration in organisms that have mitochondria comes from that last step. So we still can get some ATP from glycolysis and citric acid cycle. And that's called substrate level phosphorylation, is the method that we use to produce the ATP. So for each molecule of glucose that eventually is going to end up as carbon dioxide and water, the cell is going to make up to 32 molecules of ATP. So substrate level phosphorylation means that basically we have an enzyme that can take a substrate with a phosphate group on it and add that onto ADP to produce ATP. So that's what we mean by substrate level phosphorylation. We're taking a phosphate group off of one molecule and adding it to ADP. And we'll see that oxidative phosphorylation is different. It doesn't quite work the same way. Okay, so it all starts in every organism, whether it has a mitochondrion or not, with glycolysis. So glycolysis, as I said, it's a universal process. Bacteria do it. All eukaryotes perform glycolysis. Archaeans do it. It is one of these things like DNA, like having ribosomes. Every living thing has glycolysis as a way of producing ATP. And what glycolysis does is it breaks the sugar in half, and eventually you end up with two molecules of a 3-carbon substance called pyruvate, or pyruvic acid. So pyruvate is the name for the ion. It has a carboxyl group on it, so it's actually an organic acid called pyruvic acid. So pyruvic acid, pyruvate, essentially the same thing. Glycolysis happens in the cytoplasm, and there's two phases that we need to know about, which are the energy investment phase and the energy payoff phase. And as you'll see, glycolysis happens regardless of whether there's oxygen or not. So there's a lot of organisms that do not grow in the presence of oxygen, and glycolysis don't care. As you will see, there's no place in glycolysis where we need to add oxygen. We're not going to find out what happens to oxygen, that the oxygen that we breathe in, so essential to respiration, until the very end. Okay, so this is another one of these slides which I would call very important. It summarizes a lot of information, which I'm going to tell you about all of the steps in glycolysis. But here's our energy investment phase. We start off with glucose, and then we need to expend 2 molecules of ATP. So that's the energy investment. We actually hydrolyze 2 molecules of ATP to get 2 molecules of ADP plus 2 phosphates. So the energy payoff phase, we get 4 ADP and 4 phosphate groups, and we produce 4 ATP. So we've doubled down on our investment, as you can see. But also, we get 2 molecules of NADH. So we've harvested 4 electrons from our glucose molecule, or what's left of it. So we're going to use that reducing power later on. So all of that's going to come in at the end. So you can probably glean that NADH has something to do with what happens to oxygen. So those electrons, they get on the electron bus to go to electron camp, and we end up with 4 ATP, 2 NADH, and 2 molecules of what's left of glucose, which is pyruvate, and some water. So what goes in? Glucose goes in, pyruvate comes out, ATP is formed, but ATP is also used, but our net is 2 ATP. So we've doubled down on our energy investment. We also get 4 electrons, 4 protons that are added onto 2 molecules of NAD+, the electron bus. So we get 2 molecules of NADH and 2 protons. All right, so what's happening in gory detail? Now I'm going to tell you this, and when you take biochemistry, chances are your biochemistry professor is going to make you memorize all of this. But for now, I'm just going to show you how it is a multi-step process catalyzed by several different enzymes. This is not so important, but listen up anyway, because it's interesting. And, this is what's keeping you alive right now. So we start with glucose. And the first thing we do is we add a molecule of ATP. We add a phosphate group to it. We form this intermediate called glucose 6-phosphate. So we have a phosphate group on the 6th carbon in our glucose. That is catalyzed by an enzyme called hexokinase. So hexo comes from hexose, because glucose is a 6-carbon sugar. Kinase comes from kinase like kinetic energy, because we're adding energy to it. Kinases tend to be enzymes that phosphorylate things. We then rearrange the carbons in glucose 6-phosphate using an isomerase, which makes fructose 6-phosphate. Remember, glucose and fructose are isomers, structural isomers, so we rearrange our glucose a little bit and we end up with fructose 6-phosphate. That's catalyzed by phosphoglucoisomerase. We add another molecule of ATP. We phosphorylate this fructose 6-phosphate to get fructose 1,6-bisphosphate. You can see it's got 2 phosphate groups on it. That's catalyzed by phosphofructokinase. And then we get to break This is where the breaking occurs through an enzyme called aldolase, and we get glyceraldehyde 3-phosphate and DHAP, which is dihydroxyacetone phosphate. So we get two 3-carbon molecules with a phosphate group on them. And these two molecules can be converted back and forth to each other with an enzyme that's just called isomerase.